Hello everyone. So the next topic which we will be going to discuss is about the imbibition. So in this we will be discussing about what is this process, what are the factors that affect this imbibition, then how it plays a role in the plant cells, and what is the importance of studying this, and what is the diffusion between what is the difference between imbibition and the diffusion process. So let's start with the first slide of imbibition. So introduction part. So imbibition is a type of diffusion where the water is absorbed by the solid particles called the colloids, and they cause an enormous increase in volume. The solution is not formed in the process. In other words, water absorption by colloids is known as imbibition, and colloids are hydrophilic in nature. So in this particular diagram, what is shown that there are dry seeds. These are the dry seeds. We have taken two beakers. First is beaker 1 and the second one is beaker 2. So in the first beaker, there are dry seeds, water and the beaker. So we have soaked these seeds in the water for a long time, and in the next day or after some time, the seeds get swollen up. So this is the process of imbibition where the water is being absorbed by the solid particles called colloids, and there is an enormous increase in the volume. So the volume has increased in this case. There were dry seeds, but the volume has not increased because they were soaked in water and they have not absorbed the water. But in case of the second beaker, the water is being absorbed by the dry seeds and they get swollen up. So the solid substances are referred to as imbibents, and Im the embedded liquid is called as imbibate. So the liquid is called imbibate, and the seeds that got swelled up, it is called imbibents. So example, the absorption of water by seed or dry wood. This is the example. Then the capacity of imbibiting will differ in different imbibents. For instance, consider proteins. Since it is hydrophilic colloids, it will have maximum imbibing capacity, and compared to this, starch has less capacity and cellulose will have the least capacity. So this was about the process, and this was the experiment which was being done on the process of imbibition to understand this process better. Next is, what is imbibition theory? How this theory came up into effect. So there was a scientist, a German botanist in the 19th century. He works on the movement of water in plants and has influenced the further studies which is related to plant physiology. So what this German botanist do, he on plant physiology has described the process of the movement of water from the soil to other cells of the roots through the root hairs. So he explained this movement of water in case of the plants. So he postulated this imbibition theory in 1874, which stated that the water does not move within the cell cavities but in tubes in the wall of the plants. So they will move within the cell cavities but in the tubes in the walls of the plants. So there are many other proposals related to plant physiology by Sachs. He also proposed that the chlorophyll does not generally diffuse to all the tissues of the plants, but it contains some special entities of the cells later called the chloroplast. So it was later named as the chloroplast. He was the one person who proved that the starch formed during the photosynthesis was due to the absorption of the CO2 by the plants, and also that the starch is the first product which is formed during the photosynthesis. So he has given a big experiment on imbibition theory. He was the one who told that the starch was the first product to be formed during the process of photosynthesis. He also termed the chloroplast term, and he described the movement of water from the soil to other cells. So this was all about the imbibition theory. Then what are the factors that leads to the that leads to affect the imbibition? So water potential gradient, there is absorption cavity of imbibin, the affinity between imbibant and imbibate, the pH of the system, the temperature of the system, the pressure of the system, the cavity of the imbibant to increase the volume, and the heat of wetting. So all these factors we will be discussing in detail in these slides. So first we will talk about the water potential. So imbibants, they must have very high negative potential, water potential. So the more negative is the water potential of any substance, the greater is its capacity to imbibe. Water is considered to have the highest water potential, which is equal to 0. So water is considered to be the highest water potential, which is equals to 0. So this is about the water potential which is affecting the imbibition condition. Then the next factor which we will be discussing is about the water potential gradient. Water moves from a higher potential to a lower potential. So the drier the imbibant is, the steeper is the water potential gradient. The water potential gradient determines the rate of imbibition. So there's a water potential gradient which helps to determine the rate of imbibition. Then the absorption capacity of the imbibant. There's an absorption capacity of the imbibant which depends upon the hydrophilicity, and it is defined by the degree of attraction that the imbibant has for water molecules. So it determines how much water can diffuse into the body and can be held by the imbibant molecules. So it tells this condition of absorption capacity of the imbibant. Then next comes the affinity between the imbibant and the imbibate. The imbibant needs to have an affinity for the imbibate for imbibition to occur. So if the imbibate molecules repel, the molecule of the imbibant imbibition will not occur. So this was how the affinity of imbibant and imbibate affects the imbibition. Then the pH of the system, it plays a major role in the process of imbibition because it determines the affinity of the imbibant for the imbibate. So it is most crucial. Then the temperature of the system, it basically determines the molecular spacing as well as the energy per molecule of any system. So when there is an increase in temperature, the molecular spacing of the imbibant increases and thus increases the chances and extent of imbibition. So it will increase the chances and effect of the imbibition. The pressure of the system. The pressure also determines the molecular spacing of any system. So therefore the pressure of the system determines the imbibition rate. So when there is a pressure between the molecular spacing of the system, it determines the imbibition rate of the system. Then the capacity of the imbibant to increase the volume. So the imbibant must have the capacity to increase in volume after the imbibition process. So if the imbibant has a rigid outer layer, it reduces the ability to increase in volume, and as a result the rate of imbibition will decrease. Now the last factor is heat of wetting. So heat is produced during the process of imbibition. So this heat is called the heat of wetting. Now next we will discuss what are the conditions that are necessary for the imbibition to occur. So there should be a water potential gradient which must be created between the imbibate and the imbibant. So the imbibant moves from a region of higher water potential to a region of lower water potential gradient. So this is the first condition that is necessary for the imbibition to occur. The second necessary condition is there should be some attraction of philicity between the imbibant and the imbibate. This is the second most important condition. And third is when there is an increase in temperature, it increases the rate of imbibition. So we can say in this case that the temperature and the rate of imbibition is directly proportional to each other, because as the temperature increases, there will be an increase in the rate of imbibition. So it will be directly proportional. So these are the three main conditions that are necessary for imbibition to occur. Next is what is the role of imbibition in plants. So how does imbibition play role in plants? So imbibition has important role to play in the process of plant development. There is a seed germination and initial stages of plant growth. Then seed germination begins with the breaking of the seed coat. So when a seed germinates, it breaks its seed coat and a radicle comes out of it. So here when the seed germination begins. So water is embedded when the seed coat is broken. So water will enter when the seed coat is broken. So after the seed coat is broken, the seedling grows. So seedling will grow and needs to ascend through the soil above it. So it will, when the plant grows, you have seen that when the plant grows, it will be above the soil. So after the seed coat is broken, the seedling will grow and it will move towards the soil and it will be grown above the soil. So the growth of seedling has a contribution from the imbibition pressure developed within it. So this is the contribution in which the imbibition pressure is being developed in it because of the growth of the seedling. So the absorption of water by the roots, imbibition role comes in the absorption only. So here what is happening when there is an absorption of water by the roots? So roots are the main tissues which is required for collecting water from the soil. So the water moves into the root here by imbibition, and imbibition plays a major part in the ascent of sap through the plant body. So the plant keeps the cell moist by transferring water from the adjacent cells through the imbibition process. So this is all about the role of imbibition in the plants. So what is the significance of imbibition in nature? Why are we focusing on imbibition so much? Because imbibition helps in the seed germination. First step is imbibition, they helps in the seed germination. Second is they help in the water absorption by the root hairs of the plants, and third is they help to keep the cells moist. So these are the three main significance of the imbibition in nature in case of plants. Then imbibition is also a type of diffusion. So what is the difference between imbibition and the diffusion process? So diffusion, they involves the movement of any substance from a region of its higher concentration to a region of lower concentration. But in case of imbibition, there is a movement of solvent from a region of higher concentration to a region of its lower concentration. So there is generally a movement of water. Then there is no adsorption that is involved. There is only adsorption in case of imbibition. There is only adsorption which is involved. Then in case of diffusion, there is no change in volume, but in case of imbibition, the volume of the imbibant increases. In case of diffusion, there is no affinity which is required, and in case of imbibition, there is the affinity which must be present between the imbibant and the imbibate. So we have studied all the factors that it will depends on the volume, it will depends on the heat wetting, it will depends on the affinity between imbibate and imbibant. So these are all the things which are needed for the imbibition. Then little change in pressure in case of diffusion, but there is an enormous increase in the pressure in case of imbibition. In this, no heat is liberated. In case of diffusion, there is only a movement of a substance from a higher concentration to lower concentration, and no heat is liberated. But in case of imbibition, there is a heat which is being liberated. Then there is diffusion of gases which can also take place, but in case of imbibition, there is no diffusion of gases which takes place. So there is no gas which has been involved in the case of imbibition process. So this was all about the imbibition and the difference between the diffusion and the